Welcome, everyone. Thank you for joining us for today's webinar, Flow-Through Polishing and the Use of Multi-Mode Ligand Libraries to Improve Process Efficiencies. It is presented by Buzz Lobbezoo, the Product Support and Development Manager at Prometic Bioseparations. My name is Judy O'Rourke, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by Labroots and sponsored by Prometic Bioseparations. For more information about our sponsor, visit www.prometicbioseparations.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want during the presentation. Simply click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box. Our speaker will respond to your questions via email following the presentation. If you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're experiencing a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Now, without further ado, please join me in welcoming Buzz Lobbezoo. I will now turn the presentation over to him.
Thank you for that. Hello, everyone. As said, my name is Buzz Lobbezoo, and I'm the Product Support and Development Manager at Prometic Bioseparations Ltd. I've been with the company for the last 14 years, and in that time, I've carried out downstream processing, troubleshooting, and working with our products as well as our customers alike. I would just like a short moment to mention a little bit more about Prometic Bioseparations, who we are and what we do. Prometic Bioseparations Ltd, or PBL for short, is an enabling technologies company providing a range of products and services to the biotech and biopharmaceutical industries for the last 30 years. PBL supply a wide range of bioseparation products, both off the shelf as well as custom through our custom development programs. We have the capacity to produce over 1,000-liter batches sizes, and our adsorbents have been used in the FDA, MEA licensed products and processes. PBL also provides the MMN plates for rapid screening applications, as well as a range of columns, both standard and disposable. Today, I'd like to review the use of multimode ligands and their application in the flow-through polishing steps. Firstly, I'd like to go over the types of purification techniques there are. A number of different methods have been developed over the years to separate protein mixtures and deliver a purified protein product. These methods vary widely in their selectivity and the resulting purity of the target protein. Differences in size can be exploited to resolve protein mixtures. However, gel filtration or size exclusion chromatography has now been largely superseded by membrane separation techniques. Ion exchange chromatography and hydrophobic interaction chromatography, or HIC for short, feature in many downstream processes and separate proteins on the basis of differences in net charge or polarity. These methods remain very useful techniques in biomolecule separations, but can be limiting when the products and the impurities have a similar charge and/or hydrophobicity. More recently, ligands combining both ionic and hydrophobic groups have been developed, the so-called mixed mode or multimode ligands, which provide an increase in selectivity and tolerance for process buffer composition in comparison to ion exchange or HIC alone. Finally, affinity chromatography remains the most selective purification technique and utilizes selective binding interactions between an immobilized affinity ligand and a specific binding site on the target protein. Looking back over the last 30 years, downstream processes have changed significantly. Traditionally, purification processes consisted of a series of steps which provided progressive increases in purity until eventually the desired purity was achieved. However, this approach involves many unit operations, which contributes to the increased costs and time, as well as reduced yields. More recently, modern downstream processes are making use of an affinity capture step to provide a large increase in purity at the first step. This makes the removal of residual impurities relatively straightforward, by using one or more polishing steps where the impurities are captured and the product passes straight through. Today, as an example, most monoclonal antibodies are purified using this approach, using immobilized protein A as the affinity capture step, followed by a couple of polishing steps. Multimode ligands have grown to prominence in recent years due to their usefulness as general ligands for capture and polishing applications. These ligands combine a charged group and a hydrophobic group into one ligand structure. The example shown here is GE Healthcare's Capto-MMC ligand. Binding and elution is performed by tuning the buffer conditions to favor protein absorption or desorption. This is achieved by the addition or removal of salts and/or the addition or removal of polarity-reducing agents such as alcohols or glycols. However, there is currently a significant limitation to the application of multimode ligands in that relatively few of these ligands are currently available commercially. As a consequence, buffer and loading conditions need to be optimized to suit the ligands available, which may not always be compatible with the protein in question. We believe a better approach to multimode ligand chromatography is to produce a diverse array of multimode ligands and select the ligand that has the binding and elution properties compatible with the target protein and feedstock. In view of the limitations of current multimode ligands, PBL has developed a multimode mimetic ligand library, or MMM plate. This library consists of 96 different ligands attached to agarose beads via a spacer arm. Each ligand has a different composition of charge and hydrophobic groups. Both anion and cation groups and aliphatic and aromatic hydrophobic groups are incorporated into the library. The plate itself consists of a block of 96 microcolumns. Each column has a packed volume of 0.25 ml or 250 microliters and are arranged in a conventional 12 by 8 array format. This enables rapid screening to identify the ligand which is best suited for capture or polishing of the target protein without the need to adjust or optimize the starting protein solution. Because of this increased diversity of the multimode mimetic ligands, we believe that their selectivity is higher than conventional mixed-mode ligands. So we see ourselves sitting between multimode ligands and affinity capture. The multimode mimetic ligand libraries are created by sequential addition of charge and hydrophobic groups to a triazine core structure. This chemistry is very controllable, reliable, and gives rise to highly stable affinity ligands that can be cleaned using sodium hydroxide. A wide variety of charge groups and hydrophobic groups are incorporated into the multimode mimetic ligands. Some examples are shown here. For instance, in the ionic range, sulfonates, carboxylates, phenolates, amino, pyridyl, amidino, and so on, and the hydrophobic is alkyl, phenol, and cyclohexyl. This range provides the broad diversity of the ligand binding properties. The multimode mimetic ligand library, or MMM plate, is organized into zones with ligands having broadly similar properties clustered together to simplify selection of binding and elution conditions. Zone 1, on the left, contains ligands with predominantly ionic groups, so binding will occur at low ionic strength and elution at high ionic strength. Zone 6, on the right, contains predominantly hydrophobic ligands, so binding will occur at predominantly high ionic strength and elution at low ionic strength. And the zones in the middle, zones 2, 3, 4, and 5, contains mixtures of ionic and hydrophobic groups organized into predominantly anionic, zones 2 and 4, and predominantly cationic, zones 3 and 5, groups. The aliphatic hydrophobic groups are gathered together in zones 2 and 3, and the aromatic hydrophobic groups are found in zones 4 and 5. To demonstrate the diversity of the MMM plate, the chemical composition of each ligand can be analyzed with respect to various physical or chemical properties. The distribution of hydrophobic and hydrophilic accessible surfaces is shown in the upper two plots. These descriptors are described using the Schrödinger suite software. As expected, there is significant variation of hydrophobic and hydrophilic regions that correlate with ligand structure. Likewise, a similarly broad distribution of hydrogen bond donor and acceptor groups is displayed, as shown in the lower two plots. Other properties can be analyzed in the same way, such as polarity, aromaticity, and flexibility, as well as predicted binding to proteins. It's interesting to note that the similar patterns are displayed for polarity, flexibility, and binding to albumin. This is a consequence of the propensity of albumin to bind to long flexible hydrocarbon chains, or fatty acids. Consequently, analysis of these chemical descriptors is a useful way of confirming library diversity. This broad ligand diversity means it's now possible to select a multimode adsorbent with properties directly suited to the purification in hand. Due to its ease of use, the screening and adsorbent selection can be performed very simply in a couple of hours without the need for expensive and complex automated liquid handling systems. The plates are comprised, as mentioned earlier, of 96 discrete packed mini columns with top and bottom frits and will freely drain under gravity, so automated liquid handling systems are not required, but could be used if available. Otherwise, a multi-channel pipette is all that is required. After equilibration, the sample solution is applied to all the columns and the flow-through, wash, and elution fractions are collected by drainage into deep well collection plates. Collected fractions can be analyzed using a variety of plate assays to measure the amount of target protein and contaminants in each fraction. This allows for the rapid identification of ligands which bind the target but not the contaminants, as required for capture applications, or ligands which bind contaminants but not the target, as required for flow-through polishing applications. As an example of the use of plates, a CHO feedstock containing a monoclonal antibody was loaded into each column of the library, which was pre-equilibrated with a phosphate buffer matching the pH and conductivity of the feedstock. After loading, all the columns were washed with equilibration buffer to remove any non-bound material, and the elution was carried out with a low pH sodium citrate buffer. The conditions and descriptions and applications are shown here. Analysis of the elution fractions showed a broad spectrum of binding ranging from 0 to almost complete binding of the loaded antibody. Zone 2 in particular contains ligands which demonstrated high antibody binding as well as ligands which exhibited little or no antibody binding. This is highlighted in the orange box. SDS page analysis of eluted fractions from zone 2 of the plate reveals some ligands bind significant amounts of host cell protein and protein-related impurities. In particular, ligands C3, C4, C5, and D3 through D6 bound large amounts of host cell protein, but displayed very little affinity for the whole antibody. Consequently, these ligands were of interest for the development of a flow-through polishing step for host cell protein and product-related impurity reduction for this antibody. Analysis of elution fractions for host cell protein, or HCP, using time-resolved fluorescence assay, or HTRF, reveals several ligands binding significant quantities of CHO host cell proteins, particularly the ligands in rows C and D, confirming the SDS page findings. Comparison of the results for the elution fractions using different detection assays reveals 3 ligands exhibiting low antibody bindings but very high binding of host cell protein, as highlighted with the yellow boxes. Whilst these ligands, C2, C3, and C9, bind significant amounts of host cell protein, they exhibit very little binding of product-related impurities. However, other ligands, such as A11, B11, and D9, highlighted in green, demonstrated very low binding of whole antibody, medium to high binding of host cell proteins, and high binding of product-related impurities. Consequently, by screening the multimode mimetic ligand library, it is possible to identify and select multimode ligands for the binding and removal of specific problem impurities from the whole antibody without the need to adjust the applied feedstock. Summary comparison of STS page and host cell protein results for the 6 selected ligands is shown here. All 6 ligands display very low binding of the full chain antibody, which makes these ligands very well suited for use in flow-through polishing applications for monoclonal antibodies expressed in CHO cells. Moving on to other feedstocks, this example highlights the use of the MMM plate to identify ligands capable for the flow-through polishing of intravenous immunoglobulin solution, or IVIG, by capturing and removing IgA. 6 ligands, highlighted with orange boxes, were identified which gave more than 95% binding of the IgA with little to no binding of the IgG. Turning now to a more complex medium, human plasma, which contains many thousands of different proteins, a number of which are of interest as a therapeutic product for use in protein replacement therapies. The issue of protein impurities in the context of human plasma purification is interesting, as all the proteins are native to the human. However, depending upon the application for the purified plasma protein, certain protein impurities are undesirable and need to be reduced to very low concentrations. For example, activated clotting factors are of a concern, as these can result in thrombolytic effects if inadvertently administered in significant concentrations. This is assuming, of course, that the product is not a clotting factor concentrate. Likewise, activated proteases can result in uncontrolled bleeding episodes. Reducing the concentration of undesirable plasma proteins to acceptable levels can present significant challenges and has resulted in plasma product recalls in the past. Consequently, the MMM plates have been used in a flow-through mode to identify ligands capable of removing specific problem impurities from the product protein. In this example, the MMM plate was screened for ligands which bind kallikrein but not IgG. IgG in this case being used as the marker protein. The MMM plate was equilibrated to the same conductivity and pH as that of plasma, and 1 ml of plasma was loaded onto all the columns. The plate was washed to remove the non-bound material, and a general elution condition of a neutral buffer containing 1 molar salt and 10% hexanediol was applied to all the columns. Although several hits were observed which bound kallikrein, especially in zones 5, and that's denoted in a more red color, one ligand was identified, G1, highlighted in yellow, which bound 87% of the kallikrein present, but exhibited little to no binding of IgG. In another example, an MMM plate was screened for ligands capable of removing factor 11 from plasma. The same load and conditions were used as before. Again, several hits were identified, but 3 ligands, highlighted again in yellow, were of further interest, which gave over 85% factor 11 removal with little to no binding of IgG. As with kallikrein, G1 was identified as a hit. To verify the results of the library screens and to demonstrate the comparability of the MMM plate columns to more traditional columns, adsorbent G1 was selected for a 10-fold scale-up to a 2.5 ml borosilicate column, which was loaded with 20 ml of plasma. This resulted in a classical affinity-style purification profile with target plasma proteins passing through the column unhindered. Analysis of collected fractions demonstrated greater than 98% of the IgG being recovered in the flow-through fraction. The adsorbent G1 bound 94% of the factor 11 present, which was subsequently recovered in the elution fraction, along with several other minor proteins. And that can be seen with the STS page. So in conclusion, we have shown that the multimode mimetic ligand library, or MMM plate for short, is a useful tool for rapidly identifying adsorbents for flow-through polishing of the target proteins by removing specific protein impurities. This can be achieved without the adjustment of the starting feedstock, and the diversity of the ligand library allows for the individual ligands to bind proteins under a wide range of buffer conditions. Once a hit is identified, PBL can provide larger quantities of the selected adsorbent for further development and scale-up studies, or initiate a further ligand development program of the adsorbent. Thank you for your time and attention, and I would be happy to answer any questions you may have.
Thank you, Buzz, for that informative presentation. I would also like to thank Prometic Bioseparations for making today's educational webcast possible. Before we go, I want to let everyone know that this webcast will be available for on-demand viewing through April 30th, 2019. As a final reminder, our speaker will follow up with any questions you've submitted via email. That's all for now. Thank you for joining us, and we will see you next time. Goodbye.